children, and welcome to the new session of biological science topic nutrition, the food supplying system. Today we will discuss about the previous topics which we discussed in the part 1, that is, do you think photosynthesis is important? Photosynthesis is important as the food material is prepared in the process of photosynthesis and plants perform the process of photosynthesis. That is why we call plants as universal plant providers. Do colored leaves perform photosynthesis? Colored leaves do not perform photosynthesis because they are red brown in color and they also have red brown pigments, whereas green colored leaves perform photosynthesis because they have green colored pigment, that is chlorophyll. In the last class we discussed about methylated spirit in starch test. So in green leaves there is green color pigment called as chlorophyll. To remove that chlorophyll we use the chemical methylated spirit. So that is the importance of methylated spirit in the starch test. Today's main topic which we are going to discuss is about Joseph Priestley. He is 1733 and 1814. He is the scientist who discovered oxygen in the year 1774, whereas the scientist Lavoisier in 1775 coined the term oxygen. Relation between rat, light and mint plant. The experiment conducted by Priestley in bell jar, we call it as bell jar experiment. In a bell jar he placed a rat and a burning candle. After some time Priestley observed that the rat died and even the burning candle was put off. After some time he placed a plant, a rat and a burning candle. Priestley's experiment concluded that gaseous exchange was going in the plants and they give out gas that supported burning and was essential for the survival of animals. So in this experiment plants released oxygen and the oxygen was used by the rat and the burning candle also burnt without any problem. So plants restored the gas that was given out by the rat and the burning candle. Next activity is carbon dioxide is necessary for photosynthesis and we call it as Mole's half-leaf experiment. So Mole's half-leaf experiment is to prove that carbon dioxide is necessary for photosynthesis. The materials required for Mole's half-leaf experiment is a glass bottle, a cork and potassium hydroxide. This is the setup of the activity. Take a healthy potted plant and take a leaf, place it inside the bottle in such a way that half of the leaf is outside the bottle and half of the leaf is inside the bottle. Inside the bottle we place a little amount of potassium hydroxide or KOH and we will close the bottle tightly with the help of the split cork and we also apply some grease or Vaseline so that there will not be any gas entry or exit inside the bottle. After keeping the plant in the sunlight for 2 to 3 hours, we remove the leaf which is inside the bottle and conduct the test. For this, when we apply methylated spirit, we observe that the leaf part which was exposed to the atmospheric air and light become bluish black and the one inside the bottle containing potassium hydroxide which absorbs carbon dioxide in the bottle remains colorless. That means the part of the leaf which was exposed to the sunlight turned blue black, whereas the part which was inside the bottle which did not have carbon dioxide did not show any color change. So if the carbon dioxide gas is present, the color will change to blue-black. If the carbon dioxide is absent, the leaf will remain colorless. What are the precautions? While placing the leaf inside the bottle, make sure that leaf does not touch KOH. That means the leaf should be above the potassium hydroxide. The bottle should be kept airtight by applying grease or Vaseline. The next activity is oxygen is produced during photosynthesis in the presence of light. For this activity, the apparatus required are a glass beaker, a test tube, hydrilla plant which is a hydrophytic plant, a funnel which is having a wide broader part and a narrow part. Procedure: take the glass beaker, fill it with water, insert the funnel kept with hydrilla leaves inside it in upside down, that is, placing the broader side of the funnel towards the lower side and the narrow part of the funnel towards the upper side. Take a test tube, place it over the narrow part of the funnel. Leave this setup under the sunlight for 2 to 3 hours. After 2 to 3 hours, we observe gas bubbles being collected at the upper part of the test tube. So, the gas bubbles which are collected in the upper part of the test tube are nothing but the oxygen gas released by the hydrilla plants. Remove the test tube from the funnel, close it with a thumb and keep it aside. Take a burning matchbox, keep it near the mouth of the test tube. We see that the burning matchstick burns brilliantly, showing that the gas present inside the test tube is oxygen, proving that during the process of photosynthesis, oxygen gas is released. Precautions to be taken during the activity of hydrilla leaf experiment. While inverting the test tube on the funnel, ensure that the level of water in the beaker is above the level of the stem of the inverted funnel. While taking out the test tube from the funnel, close its mouth with your thumb to preserve the collected gas, because the gas may escape from the test tube. So, we have to close the test tube with the help of our thumb. Next activity: sunlight is essential for photosynthesis. Now, to prove that sunlight is essential for photosynthesis, we conduct black paper experiment. Raw materials needed for black paper experiment, or we also tell the apparatus needed for black paper experiment, are the black paper, the test tube, a potted healthy plant, iodine solution and clips. Procedure: take a potted plant, take a broadleaf, place the black paper which is cut in some shape. Maybe in the diagram we are having S, and the black paper is fixed to the leaf with the help of some clips. We are going to place this entire setup. After 2 to 3 hours, the leaf with the black paper is separated from the plant and it is done for the test. We remove the black paper, apply the iodine solution. We observe that the black paper which was covering the leaf did not turn into blue color, whereas the shape which was exposed to the sunlight turned to blue color, showing that light is important for photosynthesis. What are the precautions we have to take for this activity? The black paper strip should be attached tightly on both sides of the leaf so that no sunlight enters in the covered portions of the leaf. What we have learnt: Van Helmont found that water is essential for the increase of plant mass. Joseph Priestley in 1770 performed a series of experiments that revealed the essential role of air on the growth of green plants. We also discussed that Priestley was the person who discovered air. Carbon dioxide is essential for photosynthesis. We conducted the activity carbon dioxide is essential for photosynthesis. Next, a Dutch scientist, Jan Ingenhousz, noticed that plants release oxygen in the presence of sunlight. Engelmann detected the point of maximum rate of photosynthesis in red and blue lights. Improve your learning. That is, this is the work which you have to do after the session is over. Why do we keep the plant in dark for 1 week before conducting the experiment? Why do we use KOH in the bottle for moles half-leaf experiment? Why aquatic plants should be selected, especially the hydrilla experiment? What kind of leaves you will choose for studying photosynthesis process? We have discussed few activities in the initial topic. That is, why do we keep the plant in dark for 1 week before conducting the experiment? We keep the plant in the dark for 1 week before conducting the experiment is de-starching the plant. What is de-starching the plant? The starch prepared already in the plant will interfere in the experiment and change the end result. That is the reason we keep the plant in dark for de-starching. And in moles half-leaf experiment, we use KOH in the bottle for moles half-leaf experiment. Why? Why do we use KOH in the bottle? Because the potassium hydroxide, that is KOH, has the tendency to absorb the carbon dioxide which is inside the bottle or inside the leaf also. Why do we use KOH in the bottle for moles half-leaf experiment? Because we use KOH in moles half-leaf experiment as KOH has the capacity to absorb carbon dioxide inside the bottle or any traces of carbon dioxide present on the leaf, to absorb the carbon dioxide in the bottle. Why aquatic plants should be selected for hydrilla experiment? Aquatic plants are also called as hydrophytes. Hydro means water, phytes means plants. The plants which are present inside the water are aquatic plants, and for this experiment we have selected hydrilla leaves. To capture the oxygen gas released in the experiment, it is collected in the water, whereas for terrestrial plants oxygen is released into air. In this activity we have conducted a test by burning the matchstick and keeping near the test tube. The matchstick will burn brilliantly. So, because we have done this experiment inside the aquatic plants, we were able to collect the oxygen gas in the test tube. But if we do it in terrestrial plants, we cannot do the test as the air will combine in the atmosphere and we cannot perform the test. That is why we do the activity with aquatic plants. What kind of leaves you will choose for studying photosynthesis process? To study the process of photosynthesis, we use herbaceous plants which are thin and broad leaves. To perform the process of photosynthesis, we always talk about a green color pigment which is called as chlorophyll, and this chlorophyll pigment was discovered by Julius von Sachs. The scientist named Julius von Sachs discovered the chlorophyll pigment, and this chlorophyll pigment is present inside the cell organelle chloroplast. So, there is a difference, chloroplast and chlorophyll. Chloroplast is the cell organelle which is present inside the plant cell, and chlorophyll is the pigment. Let us discuss the transfer section of leaf. We have the upper epidermis which is single layered, and on the upper epidermis we have a thin waxy layer which is called as cuticle. It is made up of the substance cutin. Below the upper epidermis we have palisade parenchyma. Inside the palisade parenchyma we have the chlorophyll pigment, and the palisade parenchyma are closely arranged together. The intercellular spaces are very less, or even we can say the intercellular spaces may be absent. In between the leaf we find the vascular bundles, that is the xylem and the phloem. Xylem is useful for the conduction of water, whereas phloem is useful for the conduction of food material. The food material prepared in the leaf is transported to all the parts of the plant with the help of this phloem. The xylem and phloem are together nothing but the vascular bundles which we have discussed in our 9th class under ground tissues. When we go little bit down, we have spongy parenchyma. There is a difference between palisade parenchyma and spongy parenchyma. Spongy parenchyma has a lot of air spaces. There are intercellular spaces in large quantities, whereas in palisade parenchyma, the intercellular spaces are absent or very few. Now, the spongy parenchyma has lot of air spaces and there is chlorophyll pigment present in the spongy parenchyma. When we come to the lower side, we have air spaces, and on the lower side we have the lower epidermis, and on the lower epidermis we have with the single layered lower epidermis, and even the lower epidermis is covered by cuticle. Special structures which are called as stomata are present on the lower epidermis. Stomata are present on the upper epidermis as well as lower epidermis, but the number of stomata are more on the lower epidermis when compared to the upper epidermis. Let us see the layer cuticle which is present on the upper epidermis. The thick waxy layer which is present on the upper epidermis is nothing but the cuticle, and it is made up of a waxy coating, and the substance present in the cuticle is cutin. On the lower side of the epidermis, that is the lower epidermis, we find the bean shaped structures which are called as stomata. These stomata help in the exchange of gases. These stomata are especially present on the structure of the leaf. These are minute pores which are present on the leaf mainly for the exchange of gases. The palisade parenchyma, the palisade cells which are present towards the upper side of the epidermis, and these are consisting of the chlorophyll pigment. This is the spongy parenchyma. Over here a lot of intercellular spaces are there, air chamber is present, and as the stomata are present, they will help in the storage of the air in the air chambers. Let us see the structure of chloroplast. The structure of chloroplast consists of the outer membrane. We have the inner membrane, and the third membrane is the stroma. So, the structure of chloroplast consists of the outer membrane, the inner membrane, and the third membrane which is called the stroma. The green color structures which are present inside the stroma are called as the structure of chloroplast consists of the outer membrane, the inner membrane, and the stroma. Inside the stroma, we have the coin-like structures which are placed one above the other. These are called as grana. The single green colored coin-like structure is called as grana, and the grana arranged one above the other are called as thylakoids. Stack of grana arranged one above the other are called as thylakoids. The thylakoids are attached to one another with the help of stroma thylakoids. There are few starch cells which are present inside the chloroplast also. Let us discuss the structure of chloroplast once again. The structure of chloroplast has an outer membrane, an inner membrane, and the third membrane which is called as the stroma. Inside the stroma, we have coin-like structures which are called as grana, and pile of grana which are arranged one above the other are called as thylakoids, and the group of thylakoids are attached to each other with the help of stroma thylakoids. Along with the thylakoids, we have few starch cells in the structure of chloroplast. Now, let us discuss about the photosynthetic pigments. Because of the presence of these photosynthetic pigments, the process of photosynthesis takes place inside the plants. Let us see the photosynthetic pigments present in the plants. First pigment present inside the plants is chlorophyll A. Children, this chlorophyll A is bluish green pigment, and this chlorophyll A is just similar to the hemoglobin which is present inside our body. When you see the structure of chlorophyll A, we have magnesium present in the center, as the blood which is present inside our body has heme. The heme is also having the same structure like the chlorophyll, but the only difference is in human beings, in the place of magnesium, iron is present. The entire structure is just similar like chlorophyll A, but in chlorophyll A we have magnesium, but in human beings we have iron. That is why, children, when you go to a doctor and when he tell you there is less blood in your body, he tell you have iron supplements. That is why the structure of chlorophyll A is little bit similar or similar to the heme present in our body. Major difference is in chlorophyll A, the structure is consisting of magnesium, whereas in heme, iron element is present. So, coming to the pigments, there are two pigments, that is chlorophyll A, which is bluish green pigment, and the next pigment is chlorophyll A is bluish green pigment, whereas chlorophyll B is yellowish green pigment. What are the events which take place in chloroplast? We have discussed the structure of chloroplast. We have discussed the pigments, that is chlorophyll A and chlorophyll B, which are the major pigments in chloroplast. Now, the events which take place in chloroplast is the topic which we will discuss now. Children, we have three major events which occur in chloroplast, that is splitting of the water molecule, conversion of the light energy to chemical energy, reduction of carbon dioxide to carbohydrates. Let us go through this once again. The first event which takes place in chloroplast is splitting of the water molecule, conversion of light energy to chemical energy, and reduction of carbon dioxide to carbohydrate. So, events in chloroplast, that is splitting of the water molecule, conversion of light energy to chemical energy, and reduction of carbon dioxide to carbohydrate. Mechanism of photosynthesis. The mechanism of photosynthesis takes place inside the chloroplast, and it has two phases. First one is light dependent reaction and second one is light independent reaction. Light dependent reaction is called as photochemical phase and light independent reaction is called as biosynthetic phase. What we have learnt in this class, that is, we discussed chlorophyll was discovered by Julius von Sachs, and the structure of leaf, TS of leaf, leaf has upper and lower epidermis which protects the inner parts of the leaf, and we have kidney or bean shaped specialized structures called stomata which are present on the epidermis, that is upper and lower epidermis, which are useful for the gaseous exchange. And we have two type of tissues, that is palisade cells and the spongy cells. Columnar cells which are called palisade cells which contain high number of chloroplasts, and lower side of the leaf has spongy cells. Due to irregular arrangement, chambers are formed, air chambers. Chloroplast, double membrane bound organelle, contains thylakoids. Pigments are present in the lumen of the thylakoid and trap solar energy, converting it into chemical energy like ATP. Columnar cells called palisade cells which contain high number of chloroplasts in their cells. Lower side of leaf, spongy cells are present. Due to irregular arrangement of air chambers are formed. The work which I would like to give you all now is what makes chloroplast appear completely different from other organelles. In most of the leaves, the upper surface will be more green and shiny than the lower surface. Why? For better understanding of the concepts, solve the worksheets which are present in our official website of SCERT. Thank you.